Hello everyone, this is Kelly Eversole. We're going to wait just a few minutes to let everyone join. All right, well, let's get underway. This is Kelly Eversole. I'm the Executive Director of the International Wheat Genome Sequencing Consortium. And I'd like to thank everyone for your participation today, especially during a very difficult time in our world. And I hope everyone is safe and that your loved ones and colleagues are healthy and that you're able to stay safe. Before we get underway for our webinar, we were really saddened to learn about the death of Antonio Michele Stanza, or Michele as we all knew him. He was very involved in the IWGSC in the beginning, and he has passed this last weekend from the coronavirus, COVID-19. And it's a great loss to the community. He was a renowned plant geneticist in wheat and barley, and it was at the University degli Studi di Modena. Of significance, he was vice president of the National Academy of Agriculture in Italy. And as I mentioned, he was an early supporter of the IWGSC, and he helped launch the physical mapping of the bread wheat chromosome 5A, which was ultimately used for the reference sequence. And he was also the lead PI on the wheat chromosome 5A survey sequence. So it's a huge loss to our community, and just wanted to recognize the role that he has played in wheat genomics. So as many of you know, the International Wheat Genome Sequencing Consortium is a public-private partnership, and we have members in 70 countries, and we have more than 800 research institutes involved. I would like to, at this point, thank our sponsors for making it possible for us to have this webinar series. The IWGSC vision for IWGSC version 2 or phase 2 is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. To that point, some of our activities in 2020 are expansion of the IWGSC Arbor Biosciences collaboration, which we will be discussing in just a few moments. The publication of the IWGSC RevSec version 2.0, and possibly one that's called 2.1, but we haven't gotten there yet. And these are being used for gap closure of the RevSec version 1. We are also increasing the IWGSC RevSec annotation, both trying to combine manual and functional annotation, and we hope to release those sometime this summer. We are also trying to launch the IWGSC Wheat Diversity Project, where we do reference sequences of at least 8 land races that represent the breadth of wheat diversity. And then, of course, we're always encouraging pre-publication releases of genome sequences for elite varieties and other genomic resources. One of the other things we launched in 2020, and as it's turned out, it's been quite fortuitous since most people are working at home these days, is our IWGSC webinar series. So today, we're very pleased to have Jacob Inc. from Arbor Biosciences with us, and he is going to give us a a talk on the comprehensive, cost-effective exome sequencing with the Arbor Biosciences Wheat Exome Panel. So thank you for your participation, and I'm going to turn this over to Jacob.
Thanks, Kelly. Okay.
All right.
So, which screen is showing here?
I think we're getting your screen, Jacob.
Is it just one slide? Is it just a front slide?
Yes.
Okay, great. Thanks, Kelly, so much for that introduction. And again, my condolences go out to Professor Stanka's family, and of course IWGSC for their loss. My name is Jake. I lead research and development in our NGS section at Arbor Biosciences. Right now, Arbor is operating only to serve the research and materials needs for COVID-19 related activities. Most of us are working from home, but the vast majority are currently not working in a lab. We're sort of using this time to reset our priorities, take a few steps back and look at our research agenda and ask, when we get back to 100%, what are our high priority projects? And right now, just looking at the plate, the activities with the IWGSC remain a high priority for us. And like Kelly said, we're really looking forward to continuing these activities, continuing these collaborations, which have been wonderfully productive. Anyway, just to talk really briefly about Arbor Biosciences, in 2006, we were founded as Microarray by a professor and his research associate from the University of Michigan in the chemical engineering department. And they invented a DNA synthesizer, actually a microarray synthesizer. And that's why they called it Microarray, was because it was a very versatile synthesis platform that allowed highly customized designs to be synthesized quickly and inexpensively. And they sort of saw this as a great way to serve custom needs in a variety of molecular biology applications. Anyway, in 2017, we renamed our company to Arbor Biosciences because we wanted to better communicate that we really are a company devoted to serving biological research questions, not just a technology company, but instead a company really focused on genomics and answering questions about evolution and agronomy and all these applications of our technology. Anyway, we were acquired in 2019 by Chiral Technologies, which is a subsidiary of Dicel Corporation, a large Japanese corporation. It's been a wonderful transition to working with Chiral and working with Dicel, which brings us to today. And as of 2020, Arbor's technologies and consultation have enabled hundreds of client collaborator publications studying literally thousands of species, whether those are custom kits that we sent to teams throughout the world to use on whatever their research projects might be, or having done research in-house as part of our services section and part of our very collaborative research and development section. But like I mentioned before, in 2006, we were founded based on a core technology, which is this massively parallelized DNA synthesis platform. And then since 2006, the founders have gradually accrued a team of scientists with a broad range of expertise, ranging from evolutionary and molecular biology to paleogenetics and ancient DNA research, which is actually where I come from, and a lot of our leadership come from the ancient DNA world, virology, immunology, agricultural microbiology. And so what they've built over the last decade and a half has been this really core expertise of people familiar with and people who know how to execute both at the computer and in the laboratory non-model organism genomics. And using this combination of our very cost-effective DNA synthesis technology and our expertise in non-model organism genomics, really it's 3 products. We have a number of products, but there's really 3 products that form the core sort of flagship offerings at Arbor Biosciences. The largest of which is MyBaits, and this is this custom target enrichment kits that you can use in your lab on your sequencing libraries to enrich for targets of interest, no matter the organism. And then, we use MyBaits kits as part of our NGS services division, which we call MyReads. This is where we'll receive your DNAs and then send you read data after it's been converted to a sequencing library and enriched for your targets. And then we also have MyTags, which are synthetic. So these are in silico designed synthetic probes for fish for in situ hybridization. You can go to our website and, of course, read a little bit more about what those offerings entail and how they're made, and how for all 3 of these, our real core mission is to serve as sort of collaborators in your research projects. When you come to Arbor Biosciences, you talk directly to one of our scientists or multiple members of our scientific team to really serve your project's needs in a really customized fashion. Anyway, that's enough about Arbor. Let's talk a little bit about the wheat exome kit. So we have worked with a lot of researchers in the wheat community for the tenure of Arbor Biosciences' existence. We've done a lot of work with targeted resistance gene resequencing. We've done sort of mini exomes for whatever clients' applications might have been. And in 2018, we decided, we think that we could produce something really useful to the wheat community, and that was a full exome kit. And what better place to go than to the IWGSC to find out, well, what's really the gold standard? What should we use as our reference sequence? What should we consider the exome? And so we approached the IWGSC, and since then have had a wonderfully collaborative relationship on identifying where the needs are in the wheat community for targeted sequencing and for genomics generally. Anyway, so taking a look at RefSeq, if you just download the FASTA, it's about a 15 gigabase pair sequence. I understand that the genome is a little bit larger than that, but just talking about the FASTA, and of course, this is the first chromosome scale reference assembly, and it also has a wonderful annotation set. So, looking at the annotation set, it's comprised of 2 parts: the high confidence annotations, version 1.1 being what you can access right now, and this has just about 140,000 gene IDs, which comprises, if you just pull out the exons and look at, okay, how much total sequence space is this? It's about 171 megabases. And then also of real interest to people studying the wheat genome, of course, is the promoter space. And this is basically, if you just say, the 2 kilobase pairs, 5' of every exon 1 in this annotation set, you're looking at about 230 megabase pairs. Of course, it also has the low confidence annotation set, which actually has more gene IDs by my count, 165,000 gene IDs, but a little smaller space. So we're only looking at about 108 megabase pairs in total. But because there's so many genes, there's significantly more promoter space of about 260 megabase pairs. So, we wanted to know, okay, you can get this genome assembly, you can get these annotations, and we wanted to know, okay, what's really considered the exome, or if not, literally the exome, what would be the most useful thing for members of the wheat genomics community? And what we ended up doing was, with the help of the IWGSC, we simply asked that question to the IWGSC community. So what would be useful to your research? And we received about 102 respondents came back, and it was pretty clear that almost all of them thought that the high confidence exon annotation would be useful to their research. And so this was a relatively easy thing for us to identify as a great first step in developing a exome product for the community. So again, looking at the high confidence exons, it's about 171 megabase pairs. These are, chopped up into little exonic regions, as short as a few tens of base pairs up to quite long, less kilobase pair or more. I can't recall exactly right now what the upper limit is, but a variety of these loci. And what we did is to design this exome product, we looked at each individual exon. This is just an example of an exon. I'm just giving a numerical identifier here for, to describe how many exons there are. But we then padded each exon with 100 base pairs of sequence on either side. So this is, reaching into the intronic sequence a little bit. And then what we did is we used our in silico design techniques and bioinformatic techniques to propose to our system, millions and millions of probe candidates. And then for each window of 100 MER, we asked, okay, which one of these probes is best? And we identified these using our filtration methods. And what you end up with then is a subset of these candidates that are sequence composition and specificity filtered. So then, once you have this, what do you do with it? So we have this design, we synthesize these probes, and then we use them in our MyBaits technology. So just to talk about MyBaits a little bit, it's a very simple system, and many of you are already going to be familiar with hybridization capture. But what you do is you begin with an amplifiable sequencing library. So this could be an Illumina library, this could be an ion proton library. It could be a really, really long amplifiable, say, PacBio precursor library where the inserts might be up to 10 kilobase pairs. So long as it's amplifiable, it's compatible with hybridization capture. Anyway, so once you have this amplifiable sequencing library, some of your inserts are of interest. Maybe this is an exonic fragment, and then the rest of this might be intronic. Here in black, but what you do is you denature it and then hybridize MyBaits probes. And MyBaits probes are these biotinylated RNAs that land, hopefully, on your regions of interest. And occasionally you get non-specific hybridization of some kind with a big genome like wheat, highly repetitive. This is bound to happen occasionally. But the idea is then you bind these hybrids to a streptavidin-coated magnetic bead. Streptavidin and biotin have really strong affinity. These hybrids are pulled out of solution and bound to this substrate. And then you wash away, ideally, all of these non-specific hybrids or anything that didn't hybridize. And then you do a little bit of amplification, and then you send it to your sequencing platform. In the case of PacBio or Oxford Nanopore, you would then convert this double-stranded amplified library into say a hairpin Oxford Nanopore library or a PacBio dumbbell library and send that then to those platforms. Anyway, so we have this set of candidate probes. We went through this process of hybridization capture several times using wheat material that our member of our beta testing collaboration team provided. And then we went through rounds of development. And what you do is, starting with this initial alpha probe set, we tested them. And what you do is you refine then this to beta. So you do a lot of targeted sequencing, and then you ask, okay, what probes worked well, which ones didn't? Let's remove probes that are contributing significantly to off-target reads, for instance, or probes that are likely ended up not the best candidate for those loci. Let's add probes in at loci that we think maybe were not well captured, that we could redesign some of these poor candidates into better candidates. And then let's repeat the process. Let's test it again, but let's also test different parameters, let's say, different temperatures, different times, different numbers of samples pooled into individual capture reactions. And what we ended up with after beta testing was then this final kit and parameter set of about 1.7 million individual RNA probe sequences. And we determined that you can pool at least 8 hexaploid wheat samples into a single capture reaction. So when you do one of these MyBaits capture reactions, you're actually doing it on a collection of 8 samples at once, which are individually barcoded in their adapter sequence, for instance. One really key part of our development process, and which has itself turned out to be a wonderful collaboration, has been working with the Curio Genomics platform. So Curio Genomics produced this fantastic cloud-based NGS data analysis platform that you can go to their website, curiogenomics.com, and check out. But we worked closely with them during development and thereafter. And what's really cool about Curio is that it has built into it already the Chinese spring assembly and also the Svevo tetraploid assembly. When I say it's built in, it means that you can upload FASTQ data to this and immediately map to these genomes. They've already been indexed, and the mapping is incredibly fast. And of course, once it's mapped, you have this fantastic dynamic read alignment capability where, you know, navigating a whole chromosome very quickly, jumping between annotations, you know, basically at the click of your mouse is very straightforward and powerful. And also variant calling is extremely rapid, and the parameters are tunable in real time. So, you know, what Curio does is that it measures the quality of every site in the entire alignment. And then when you ask it to call variants, you can just click a different filtration set and it will instantaneously show you the results of that filtration. It's a extremely useful tool. There'll be a lot of screenshots from Curio in the rest of this talk. So I just wanted to introduce that now to you know, give you a sense of where this comes from. Anyway, so let's talk about the performance. We've talked about, you know, alpha development and then beta development. So let's talk about what this exome kit actually delivers. So with hexaploids, what we've discovered is that this kit is extremely comprehensive and specific. So the total official target space in this capture panel is 230 megabase pairs. 158 megabase pairs is explicitly exonic. You know, I mentioned it's 171 megabase pair total HC exons. This reliably covers 158 megabase pairs of that, which is over 91% of it. We currently recommend 18 gigabase pairs of paired N150 Illumina sequencing per sample. So if you're putting in a regular Illumina library, we recommend about 300 to 400 base pair inserts, and then paired N150 sequencing coupled with that. It's perhaps the least expensive sequencing that you can get right now, so we chose to spec based on that. And what you end up with is over 30x mean unique read depth across this 230 megabase pair target, pretty reliably. 97% of the exons in the high confidence exon annotation set, not merged, but just the absolute number of exons are covered by this kit. What does this translate to? Well, it translates to about a 30-fold reduction in exome sequencing versus WGS, so versus whole genome sequencing. So you're saving a tremendous amount of money getting the exome at high coverage using this kit. And looking closely at sort of the specifications, you know, it's very comprehensive. We're seeing, you know, great coverage, and you can see in these metrics, you're seeing great coverage of all the exons throughout every member of the subgenomes. You're seeing a high % on target. So the average right now is actually a little bit higher than the 70% reads on target, this blue series. And, you know, just looking at, say, an alignment plot, you know, you can see that the non-exonic coverage, of course, is an order of magnitude less or more or less than what you see in the exons. So one thing that, you know, is perhaps the best way to communicate the kinds of data that you actually get with this kit is just showing some realignments here. And, you know, whether you're surfing for mutants in an EMS line or looking for disease resistance genes, this particular plot is an NLR, just a random NLR on chromosome 7A. You know, you can retrieve at high coverage variation for basically minimal cost with this kit. You can discover major structural features of interest. Here's a site in gamma gliadin that you know, there's a clear deletion compared to the reference. These are things that you can easily retrieve and visualize very rapidly in an alignment platform like JBrowse or Curio or what have you. We knew that, you know, this kit designed from hexaploids worked quite well for hexaploids. And we, however, got a lot of questions, you know, well, what about my tetraploids? You know, what about my Durham? How does this kit work on those lines that I'm working on? And we we address this directly. So we got our hands on some tetraploid material. This is actually material that we got from Jorge Dubkovsky, and I'll mention him later. He's been a great close collaborator on this project. But what we found was that when you do a capture using this kit on a tetraploid NGS library, and then let's say you map those reads back to the hexaploid reference, back to Chinese Spring, this purple arc sort of, just to describe this really quick, if we just take a quick look at a 1 megabase pair window in Chinese Spring, these black hashes here represent exons in the genome. And we have a read coverage plot out here on the exterior of the arc. What we found was that with tetraploid capture, it's actually, the kit actually is even a little bit better than hexaploid in that you get a highly specific capture where you're getting more than 72% reads on target. And again, if you define the target here as the hexaploid exons. But of course, we were wondering, well, what about if you use a tetraploid reference? So if you just look at these sites and where we know we have probes for these sites and ask, okay, where on, say, SVEVO would these probes likely hybridize? If you just take a look at that, you might predict that probes would hybridize here in this region, et cetera. And then you ask, okay, well, what about the percent on target and the coverage of this of the exome on SVEVO on its annotation set? And we predicted just in silico that if you try to put this probe here, how much of this exome would you recover? We anticipate about 80% of that exome could be captured with the hexaploid probe set. But we were pleasantly surprised to discover that we actually got comparable exome coverage, 94% of the SVEVO exome as the hexaploid Chinese Spring exome, which is interesting. It also interesting is that I mentioned that you're getting 72% reads on target when you map tetraploid to the hexaploid reference, but it's about 59% on target for the tetraploid, at least mapping to SVEVO. But this is interesting in that still there's 40% of reads that are mapping outside of the annotated exome in SVEVO. But what's interesting is that actually this gives you wonderful discovery potential in tetraploids. So this is just a plot of, as you sequence deeper, you're, discovering more of the annotated exome space, and at 12 gigabase pairs of sequencing, you're getting, you're flattening out, but you're getting all this extra high coverage space in the SVEVO alignments. And, not only does this give you great, SNP survey within the exome itself. Here, we had, again, these 8 lines, SVEVO itself, and then also Wild-Emmer here. But you're getting, just great discovery within the exome, but also outside of the exome, you're also getting hundreds of thousands of SNPs discovered in a simple application of the hexaploid capture. So with tetraploids, we're pretty confident this is a nice kit for working with tetraploids. Of course, another question came, well, what about diploids? What about other species? So we also tried a couple diploid lines. So we tried Agelops, and you can see here when you capture Agelops with this hexaploid capture kit, you're getting, and just align, this was just for illustration, just aligning back to the hexaploid reference, you're getting these coverage peaks at its subgenome, at the right subgenome here, subgenome D. And what's interesting is that you get about 48% on target using our most recent parameter set. And you're getting really nice coverage of these exons for sure. And then we also tried this on monococcum, and this was about 45% on target using our most recent parameter set. And again, despite this significant divergence in a lot of these regions, we're still getting really good coverage of most exons with sort of minimal sequencing. So it looks like the kit probably works well for diploids. This is an active research program right now. We're looking at playing with parameters to improve the on-target rate. And I have yet to go back and actually map these back to just the individual subgenomes. This is just an illustration, but we might discover some things when we take this to phase 2. This was just phase 1 information. So great, wheat and its very close relatives, its subgenome progenitor wonderful. What about non-wheat? So we also had an opportunity to try out some barley capture using this exome, this hexaploid wheat capture. Just looking at more XV1 here. So it has an annotation set, 121 megabase per exome. I understand that a lot of that is probably lower confidence than some of it, but just looking at this, head on here, we did that same sort of in silico prediction of asking, will the probes map to how much of the exome are they likely to cover? And we predicted about 44%. And once again, we were pleasantly surprised to see that actual capture data provided, more than half of the exome covered. And we see, over 40% of the exome covered at 10x with about, pardon me, I think it's about 12 gig or 15 gig of sequencing, something like that. So it can serve as sort of a multi-taxon panel, it looks like. Again, this is another active research program that we're doing alongside our diploid captures to see how much we can capture. And then also asking the question, well, what if we wanted to make a Barley module? How much extra probe space would that include? But that's discussion for later. What about rye? We predict about 59% of that exome could be covered with this kit. If history is any lesson here, we typically see more than we predict. This is actually a great data set to improve our prediction algorithm, obviously, but we have not run these yet. If any of you out there would like to give rye a shot, please go ahead and contact Arbor, and we can talk about a potential demo there. But we anticipate that a lot of the rye genome would be covered, rye exome would be covered quite well by this kit. One question we often get is, how does this exome kit compare to others that have been developed? We know that this is not the only exome product that you can use. Ours is based on IWGSC. Our official target is a 230 megabase per target, but we know there's other designs out there. For instance, Jordan et al. In 2015 developed largely from a low copy gene locus assembly of wheat that was available at the time, and then coupling that with some transcriptome data, they developed a 107 megabase pair target set. And you can download this FASTA. It's publicly available, and you can see, it's about 107 base pairs of sequence that's comprising this target. If you chop it up into, say, 50 MER sequences and align it back to RefSeq, you get about 104 of that mapping back to RefSeq, just using that kind of crude method that I just described. And then we asked the question, with our hexaploid capture, how much of that 104 megabases is covered? And we see about 61 megabase pair you achieve at 5x using hexaploid capture at 18 gig sequencing. This is about 59% of that 104 megabase per target. Similarly, with Krasileva's 2017 exome, this is basically a large transcriptomic reference that was then mapped back to an earlier assembly of RefSeq and then padded for intronic sequence. Our kit, or excuse me, if you chop it up into 50 MER sequences and map it back to RefSeq, you get about 65 meg mapping. And we're entirely aware that this is not a perfect method of realignment. This is just to illustrate, if you apply the same technique to mapping back fragments of this target as you would read data, this is how much maps. And we see about 57 megabase pair of that covered with our kit at 5x. So that's about 88% of the target. And then most recently, a very large design, Gardner et al. Came out using IWGSC RefSeq, and then also a couple other resources, an awful lot of other resources to comprise, a really pan exomic, this high confidence, low confidence, a lot of supplementary information, supplementary sources, 518 megabase target, 450 of which is mappable using that chop technique. Again, it's entirely possible more of this would map with a different technique. But we see we cover about 238 megabase pairs of that. And you'll see that there's a contrast here between 238 and then our official 230 megabase pair target. Remember that there's 30% of sequences that are not aligning directly to this official target. And it seems that these have a beneficial impact on covering these other designs. So these are really cool designs that are available that you can go and and order and use. We think that the Arbor IWGSC design, though, is a really good balance of size, so to the sort of breadth of this spectrum, and then also knowing that we're just targeting the high confidence exons here. You get a little bit of peripheral information using the kit, which is great. So there's a lot of cross compatibility between these kits, where, you know, if you have data generated with cross-this kit, a good portion of it you're going to be able to retrieve again using our kit. So just wanted to throw that out there because I know that we've gotten a lot of questions about that. So what's next? Just wrapping up this description here. You know, we going back to that original question that we proposed to IWGSC. You know, what would be the next module that we want to include? And we understand that, you know, each individual line is going to have sections of its genome that would be of interest to an individual research program, but we definitely recognize that promoters, the promoter regions or putative promoter regions would be of real interest to everybody, and that was obvious in the numbers from this survey. So our next kit is going to be a promoters module. This would be an optional module that you can add on or just buy on its own to use in conjunction or independently of the core exome product. So what's the promoters? We have a design already made, and it's in synthesis right now. Actually, it's obviously on pause given the situation with you know, the stay-at-home order, but it's actually partially done, and we're eager to get back in the lab and start trying this out. But anyway, what are the promoters? Well, right now it's basically 2 kilobase pairs, 5' of every exon 1. But thanks to Jorge Dubkovsky, who's been a real key member of the team designing this promoters capture, we also have some open chromatin regions identified with ATAC-seq that appear to be, you know, influencing gene expression in a lot of lines. And so we have that as a supplement to this. So it's promoters plus these open chromatin regions. And then Jorge's done a lot of work using promoter capture using previous designs. And he has, you know, determined and identified experimentally, and then also other team members have identified informatically regions that are really problematic to that initial design. So in many ways, this promoter design, since we started with all this information, it's kind of already in beta, which is nice. It kind of has accelerated making this available. But anyway, the final target space is about 191 megabase pairs. It's an optional, again, an optional add-on that you could use independently if needed. And you know, like I mentioned before, you know, we've served the wheat genomics community for quite a while, for a long time before jumping into complete exome. But we intended to, you know, we realize that these are, you know, there's lots of needs for targeted sequencing outside of just an exome. But speaking of just the exome, you know, as new annotations are made available on RefSeq1, and then of course as RefSeq2 and 2.1 become available, we'll be incorporating those into the kit. We're entirely committed to making this kit as useful and up to date as possible as resources become available. We understand that there will be probably some use for sites that our kit currently doesn't cover, that are part of large SNP panels, known SNP panels, and so we'd like to incorporate those as optional modules into the kit, and so these are all planned. Anyway, I just want to, the last thing I wanted to talk about was, you know, let's put this in context. Let's look closely at, you know, what is this wheat exome kit? How much does it cost? What are you actually getting? How can you use it, et cetera? And I just wanted to wrap up with a quick description of that. So you have two options to use this exome kit. One is you can use it in your lab. And this is the classic MyBaits model. We design a kit, we send it to you, you use it how you like, and then you reap the benefits of doing the work yourself. And right now, the USD list price, and you can see this on our website, but the USD list price per sample to do this in your own lab, it starts at $37 per sample. So for $37 per sample and a little bit of work at the bench, you're saving yourself an order of magnitude or more in sequencing cost to get the exome. It's a high value proposition in our mind. Anyway, this model is you start with your gDNA, you do your own library prep, but then this is where you plug in the wheat exome kit, the MyBaits wheat exome kit, and then you take it to whatever sequencing platform you're ultimately going to take it to, whether or not that's Illumina or PacBio or Oxford Nanopore or what have you. The other option is under our MyReads umbrella. So MyReads is NGS services. You send us your gDNA, and for $5.39 per sample, again, that's starting list price on our website for hexaploids. So there's a few qualifiers there. You send us your gDNA, but then we do everything else. And then we can either send you FastQ data that then you can use on your favorite bioinformatics platform, but we're also working with Curio right now. We just found Curio so valuable during our development, and from what we can tell, it's an extremely useful platform for dealing with large genomes like wheat. But we're working with Curio right now to see if we can't put together offerings for people working with wheat exome in particular, where they can download their data directly from Curio, analyze it on Curio, visualize it, call variants, do all the things that Curio offers. So that's what you're getting here. But I wanted to mention that, you know, people need different things than just the exome, and we fully recognize that. And we've learned so much about designing probes for exomes that if you have a custom add-on that you would like to add, like maybe you have a 10 megabases of target that you would like to add on to the wheat exome. Well, those are introns or new exons, or maybe you have a big panel of NLR candidates. We're currently offering this add-on at a 25% discount against list for the rest of 2020. So if you buy the exome kit, you're planning on doing exome sequencing, we'll put this add-on, this custom add-on in there for 25% off, whether or not you're using it in your own lab or in the MyReads context. So I just wanted to mention that. So as you're at home brainstorming, you know, how you're gonna hit the ground running when you get back in, you know, back in with your team, back in the lab, back at your servers, you know, what are you gonna do? Keep this discount in mind. These are things that we can turn around rapidly and hopefully add some value to your wheat exome captures with us. Anyway, wrapping things up here, we have a lot of people to thank, but especially these people. So Burkhard Streunagel, Shreya Ghosh, Ute Baumann, Etienne Poe, and Sebastian Paul, these have been wonderful advisors during this procedure, and also beta testers of this platform. The best people I've met are in the wheat community, and these are certainly among them. And it's just been a wonderful experience working with this group of really brilliant, very clever people. And then same for our leaders on the expansion projects. So of course, Jorge, Jan, and Vijay are all working with us to figure out, okay, how do we make this exome kit as useful, as broadly useful as possible? And you know, Tetraploids, Barley, and Diploids respectively there in that list. They've been very gracious, providing materials, providing insight, and lots of advice, and making this product as good as possible. So that wraps things up for me. Thanks so much for listening to this. Check out our website. You can go direct to arborbiosci.com and just surf around. Hopefully it's relatively easy to find what you're interested in there. You could also go direct to our MyBaits Expert Wheat Exome page, where you'll see performance metrics much along the lines of what I described here. And of course, we're all about that, you know, person-to-person communication. So reach out to us at info@arborbiosci.com, and that'll get piped to a scientist who's best matched for your research needs. And hopefully, occasionally, a few of those inquiries go direct to me, because I love talking about what we can achieve with this very inexpensive synthesis technology that we have at our disposal at Arbor, and then of course the expertise that we have available too. Anyway, so that wraps things up. Thanks so much, and I guess we can turn it over back to Isabel and questions.
Great, thank you very much, Jacob. This is Kelly.
Okay,
That's okay. We do have a few questions that came in, and I don't know if you can actually see them, but I'll start with the first one. When you designed the probes, did you consider the information from other chromosomes for off-target concerns?
Yeah, absolutely. So let me run back up here. It's going to take a little bit for me to get all the way through all these animations. Right. All right. So that's in reference to this development workflow. So what we did with the alpha is we asked for each probe, how many locations in the genome is it likely to hit? Actually, it may be useful to just go up here. is it likely to hit? And that's what we call specificity. And we measured specificity in 2 ways. We asked, okay, what about off exome? And what about on exome specificity? So maybe this probe could be used to capture the same homolog in all three subgenomes. So we measured, during that probe design, we measured their efficacy both within and outside of the exome, whether or not the probe ultimately derived from, or the probe the probe could hybridize to the intended target, or then it's other sections of the exome elsewhere. So yeah, that's all part of our design and screening process.
All right, I think we have another one here, and I just had it. Lost it. With Curio Genomics.
Say that again, Kelly.
What is the mapping time with Curio Genomics?
If you load up 20 gig or 18 gig of paired N150 data, to map it to the hexaploid genome takes less than an hour and a half in most cases. And while a super large cluster could that you might have available at your institute might challenge that. What's nice is that the mapping procedure includes all of the scoring. It databases all the scoring, so that if you then want to filter that alignment by whatever feature of the BAM file, and it does simultaneous duplicate marking. It does a lot of the initial parts of variant calling also in that first alignment step. That after it's aligned, you can instantaneously turn on and off filters to look at how your alignment would be impacted if you were to remove this mapping quality or that mapping quality, or remove these PCR duplicates, or there's a number of things that you can do with it. Let's see. They have a free trial if you want to go in there and try out. We have some demo data from hexaploid captures that if you wanted to download from us, I don't know that it's posted on the website yet, but if you wanted to download it and load it up into Curio and align it, you could see pretty quickly that it's both very fast and really dynamic in how you can apply filtration.
So just so everyone will know, we did want to try to have Curio Genomics to do a webinar with them, so it may be very useful to try to do that in the next couple of months. So that may give everyone a better idea of how to use both Arbor Biosciences and Curio Genomics.
I should mention really quickly, Kelly, sorry to interrupt, but if you queue 8 or 50 samples on Curio, it scales incredibly well, almost linearly. So an hour and a half to align 12 or 48 samples, all simultaneously. You can do all the alignments simultaneously. I forgot to mention that.
The next question is, are the probes unique to one subgenome? Right,
so that's kind of related to that original question of, how specific is each individual probe? They are not necessarily specific to in sequence to the individual subgenomes. Not all of them. A lot of them are. A lot of them are very specific, but because there is sequence identity between the subgenomes, certainly in the short probe windows that we're operating in here, one probe that came from A might capture a similar locus on B, or excuse me, might have the identical sequence identity. These probes are actually, as I showed, you can capture regions of relatively high divergence with this kit. So the probes from subgenome D are likely contributing to the capture of the subgenome A loci and subgenome B loci at the same time. It's not a loose system per se, but it's highly tolerant of sequence divergence.
And then the next question is, and I believe it's time related, from capture to results prior to analysis.
Yeah, so in your own lab, you make your libraries. Maybe that takes you half a day to do half a plate of libraries. You set up the captures that night, you clean them up the next day. and then you throw them on your sequencer, and whatever your queue is then dictates how quickly you get your data. But if you, if let's say you just had a couple captures and you want to throw them on a MySeq or something like that. So day 1 is library prep and setup capture, day 2 is cleanup, and then by day 3, the end of day 3, you have data to analyze. In a MyReads context, the queue is kind of what dictates everything. But with Wheat Exome, we're typically seeing turnarounds of, as soon as you deliver your ddNA, we're seeing turnarounds 4 to 6 weeks to getting data. And if there are, time crunches and deadlines, our logistics whiz, Dr. Jennifer Klunk, is who leads MyReads, is very good at figuring out how to make things happen quickly, if things can be delivered in certain batch sizes, or if things can be spread out across multiple runs, or these sorts of things. She's very good at that. So to get a more accurate turnaround time for a given period of time, it's best to just reach out to us and see what our queue looks like.
So the next question relates to the extent to which you provide services in India, and whether, with regards to that, you also provide support for in actually analyzing the data.
Yeah, so we will sell kits anywhere. I can't remember where we sit with a distributor in India, but we are certainly capable of getting kits to India if you want to use them in your lab. As far as Curio goes, or as far as bioinformatics services go, where we currently stop right now is alignment variant calling, and then you can download both of those things directly from Curio and explore them in Curio. But beyond that, we don't have a formal bioinformatics, sort of, I don't know what level that is, tertiary or quaternary level of bioinformatics support after that basic alignment and variant calling. But that's something that we're looking seriously at now that now that we're part of the Dicel team. Dicel and Chiral both have a much larger distributor network than Arbor Biosciences ever did, and we also now have more opportunities to add on bioinformatics services potentially. It's something really interesting to us, and we think that this would be in close collaboration with Curio too.
So a follow-on to that question was how effective it would be to try to do this for 400 land races.
Yeah, that doesn't scare us. We've done very large projects. Thousands of samples is not scary for us at all. We have the capacity to do that. It's really a matter of, if you want to see, okay, what are the pricing options, turnaround time options? Those are all great questions, again, for our lead scientists over at MyReads, and all it takes is a little email or a phone call, and we can look at options there.
The next question is, when you refer to Exome Capture, does Exome include both UTR and CDS regions?
Yes, so the UTRs are included here.
Another question is, how efficient does the kit work with the long sequences, for example, from PacBio? I think you addressed that.
That is a wonderful question. We know that MyBaits kits work really well on PacBio reads, but because the panel is so large, and it's difficult to imagine a scenario where PacBio sequencing would, well, I mean, it would certainly be useful, but perhaps not the most cost-effective approach. Using a super large panel like this. So we don't actually I don't have data, that I can show with PacBio Capture using this kit, but there's a heck of a lot of literature out there showing how well MyBaits and Hybridization Capture systems works on PacBio length inserts. We've done a number of projects where we have a smaller panel that was designed for use with short read sequencing. But then when we just took that exact same probe set and applied it to PacBio inserts, we're oftentimes seeing higher percents on target with the long inserts, which is really interesting. Percent, contigs that are alignable back to the target. This is especially the case for some resistance gene sequencing applications. That's been particularly interesting to see that sometimes a panel where you might get 25 to 30% on target, if it's a really kind of rough custom panel, you might get 50 to 60% on target when you do it with PacBio, which is cool. But for the wheat exome, for this particular kit, I don't have data. If that's something that if people really want to see, shoot us an email at info@Arbor Biosciences and say, yes, I want to see wheat exome capture data for PacBio, and we'll seriously consider producing it as quickly as possible.
So to that issue, I think it would be really interesting to see it with PacBio as the sequel to.
All right. Vote. We got one vote from Kelly now. All right.
Yeah.
Excellent.
One of the other questions that came in was is Curio for free? And I already answered that. No, it is not for free. And again, that would be something we could cover in terms of cost on a separate webinar.
Right. Yeah.
And the next question is, how many variants can you typically call genome-wide for a given hexaploid wheat cultivar versus the reference sequence?
Wow, we haven't tried finding the most divergent hexaploid line to say, okay, what's the maximum number of, like, how many SNPs did we discover in this super divergent line that's been, like, say, whole genome sequenced? That's a really good question. I haven't looked directly at that. I know that with, we were comparing other panels, and Jorge might be like, Jake, you're so wrong here. But with Krasileva et al.'s panel, that 84 megabase pair one that we overlap, something like 88% of it. I think that panel, maybe Jorge's 10 million SNP panel or 10 million SNP list falls within that panel. I'd have to double check, but that's an awful lot of sites if it's just within 84 megabases. But no, that's a good question. For if we go back, or actually if we go down here to, back down here to this tetraploid example, I think against Zavitan, for Zavitan against the reference, there was something like 300,000 variants called. But as far as hexaploid, I'm not entirely sure. And my apologies if I got that description of that 10 million SNP collection that Jorge has.
Yeah. So we've reached the end of our time, but I would like to thank you very much, Jake, for a great presentation, and thanks everyone for participating in the webinar today. And this will be posted on our YouTube channel, so you'll be able to go back and look at it or share it with your colleagues. And before we have our next webinar, which we may move directly to try to line up the Curio Genomics webinar, I want everyone to please stay safe, and I hope that you and your colleagues and your loved ones will not be affected too adversely by the COVID-19. So with that, we'll close, and thank you very much.
Thank you, guys.